Hello, everyone, and welcome to the first Perspectives in FTD Research Webinar of 2022. FTD Biology and Testing. Why do you need my samples? This webinar is being presented jointly by the Association for Frontotemporal Degeneration and the FTD Disorders Registry. My name is Dr. Diana Wheaton, and I am the director of the FTD Disorders Registry. On behalf of everyone here, thank you for joining us. A few housekeeping items before we get started. Please note that the audience will be muted for the duration of the presentation portion. This helps to keep the background noise to a minimum so that everyone can hear the presentations clearly. If you have a technical issue, please write a message in the questions box, and we will try to address them with you. There will be time for question and answers at the end of the presentation. If you have a question for one of our presenters, type it in the questions box when you think of it. You don't need to wait until the end of the presentation to submit your question. We will answer as many of them as time permits. Additionally, this webinar will be recorded and archived for later viewing on both AFTD's YouTube page and the FTD Registry's YouTube page. As mentioned, I am the director of the FTD Disorders Registry. The registry is a powerful tool in the movement to discover treatments and a cure for FTD. It relies on the stories of people like you, persons diagnosed with FTD, caregivers, both current and former, family members, and friends. Your stories and experiences will help to spur innovation that will lead to effective treatments and therapies. If FTD has touched your life in some way, I strongly encourage you to join the thousands of people who have already signed up for the registry. Registration allows you to receive periodic news and FTD research updates by email, as well as notifications to participate in surveys and research studies to help further the science of FTD. A quick announcement. AFTD's annual education conference will be held Friday, April 8th, and will feature the latest information on FTD science and caregiving, as well as the opportunity to connect with others on the FTD journey. Local, state, and federal guidelines permitting, the event will take place in person at the BWI Airport Marriott near Baltimore, Maryland. AFTD will ask all in-person attendees to show proof of vaccination. The conference will also be live streamed online. Registration opens next week. Be sure to visit the AFTD website at theaftd.org for more details. Before we begin, we'd like to get a sense of your own personal feelings on participating in FTD research. Click on the answer that most accurately answers the question, would you consider taking part in FTD research in the future? We'll give you a couple minutes to make your selection. Okay, we will revisit those results later in the presentation and also give you an opportunity to respond at that time as well. Now I would like to introduce our presenters. Dr. Penny Dacks is the Senior Director of Scientific Initiatives at AFTD, where she leads strategy for AFTD's research programs. She is also President of the FTD Disorders Registry. Previously, she worked at the American Epilepsy Society, overseeing all mission-related programs in research, medical education, and clinical activities. Before that, she spent 5 years at the Alzheimer's Drug Discovery Foundation, where she led development of CognitiveVitality.org and the Aging and Alzheimer's Prevention Program to source and evaluate potential therapies. Dr. Nupur Ghoshal is an Associate Professor of Neurology and Psychiatry at Washington University School of Medicine in St. Louis and is a clinical investigator for the Knight Alzheimer's Disease Research Center. Dr. Ghoshal is the Clinical co-principal investigator for an AFTD-funded biomarkers grant that focuses on examining the production and clearance of the protein tau in progressive supranuclear palsy and cortical basal degeneration. She is also the site principal investigator for ALL-FTD, a multi-site consortium focused on studying the natural history of FTD and developing clinical trial-ready cohorts. Our third presenter is Drew, who wishes to remain anonymous. Drew has participated in FTD clinical research procedures in the past. He has graciously offered to answer some questions today for Dr. Ghoshal, who will ask him about his experiences with FTD research. Welcome to you all. And now I'll turn things over to Penny.
Thank you, Diana. And thank you to everyone for joining us today online, and to my co-presenters, Dr. Ghoshal and Drew. This is a really exciting time for FTD research. Just 10 years ago, there were few, if any, ideas for effective treatments that were ready to be tested in clinical trials. Today, there are multiple active trials, with more coming down the pike, offering hope that there could be a treatment soon for some people with FTD. Equally important, clinical researchers are mapping out how FTD happens with new technology and in more detail than ever before. And the map that they're creating, that we are all working together on, is going to help us find the path to treatments that could delay onset, slow progression, help with symptoms, and in some people even stop the disease. There are so many studies underway that some researchers are struggling to find people to participate, which is a real problem, but a very different one than the one that we had 10 years ago. So one of the reasons for excitement, for why hope is on the horizon, is actually biomarkers, which isn't something that we normally think about, but is critical to developing better care and better therapies. Which brings me to today's topic. Why do samples matter? And what are biomarkers? Why are they important? To frame it a little bit differently, why do people maybe feel like they're sometimes poked and prodded when they go through clinical research? So a biomarker is a biological marker that can measure an aspect of health or disease. And we are all used to these things. We use them all the time. From when you're a child, you go into your doctor's office, and one of the first things they do is measure your blood pressure, right? And maybe not your child, but when you're older, if that measure is not what the doctor is expecting, it's going to trigger additional evaluations. It's going to trigger additional ideas for treatment. And she may prescribe you a new treatment to lower that blood pressure. And the reason is not because of a number, right? The number is a biomarker for your risk of a stroke or heart disease. It's a biomarker, and it's one that's pretty trusted and widespread today, and it's essential to proper care for cardiovascular disease. So in FTD, biomarkers are critically needed. And in a lot of ways, it's because the brain is kind of like a black box that does not always reliably match what you see on the outside. Or framing that in a different way, the pathology or the causes of damage do not reliably match the symptoms. And I apologize because this is going to be a complicated slide, but if you look here on the right, you can see that this is more the 6 different types of clinical syndromes. Somebody who has FTD might be experiencing symptoms related to Parkinson's disease, like with CBS and PSP. They might be experiencing behavioral variant FTD symptoms. They might be experiencing language-related symptoms for the primary progressive aphasia, or they might be experiencing FTD ALS. But those clinical symptoms do not cleanly match onto a single type of damage in the brain. And that's what you can see in the middle. That green column are different types of damage that can be seen in the brain if it's expected at the time of autopsy. So you might have tau-related types of damage, or TDP-43 types of damage, or FUS. So, when scientists try to create new treatments or better therapies, particularly if they're really trying to stop the disease, then they need to go at the underlying causes of the damage. But if somebody right now creates a therapy against, say, a type of TDP-43 damage or tau, they don't know which people they need to put into their clinical trials because they don't know which people have which causes of damage. Say if they go at behavioral variant FTD, the blue circle, you can see that it's going to pretty much every single different type of those underlying causes of damage. And that reality is part of why most, not all, but many of the different clinical trials that are underway now are really targeting people who have their FTD caused by genetic causes. So most forms of FTD are not caused by genetic variants. On the left with the red circles, you can see 4 of the causes there. But most of the clinical trials happening are really looking at people who have specific genetic variants because scientists have more confidence of what does their underlying damage look like. So they have more confidence that they're finding the right people for the right treatment. So to step back, biomarkers are used for a lot of different things. One could be for accurate diagnosis. So wouldn't it be amazing if when somebody starts to experience symptoms related to depression or anxiety, their doctor could do a blood test to see if there's underlying actual damage to the brain that might indicate something like FTD versus maybe psychiatric disorders where you don't have that kind of damage. And that simple blood test might get someone from their primary care provider to a specialist who can really give them an accurate diagnosis. Biomarkers can also help match which patients have what causes of damage, i.e. which treatments do they need. It's also useful for monitoring the response to treatment. If we go back to that analogy of blood pressure, when a doctor prescribes a treatment for blood pressure, that's not the end of it, right? She monitors whether or not the blood pressure responds to that treatment. She might adjust the dosing accordingly, or she might try a different treatment if it's not working. Biomarkers are really effective at quickly getting indicators of whether a treatment is working. They can also just monitor and predict progression of disease so that you can better plan and doctors can better plan for what's going to be coming down the pike. And they're critical for accelerating drug development itself. So in any given clinical trial, a lot of times those new therapies, the first therapies tested are not going to be the ones that end up working. That's frustrating and it's maddening, but it's the reality of where we are. It's not easy to create new and effective therapies. So finding ways to use biomarkers so that those therapies can be tested more quickly, more efficiently, so that we can move past the ones that aren't working and find the ones that are. So for FTD, there's really a lot of work that still needs to be done to make sure that there are biomarkers ready for all of those different types of purposes to get better therapies and better care. You will find that there is brain imaging available, so for structural biology, right? That's a common part of clinical care today. You can figure out where in the brain somebody's got that damage. Other types of biomarkers might involve, say, PET imaging or other types of approaches where you're imaging for specific types of molecules to see their presence in the brain. The Alzheimer's disease field has seen a lot of progress in this over the last 5 to 10 years, where a doctor can order a test to see how much amyloid protein and damage is in the brain, how much tau damage is in the brain. The FTD field isn't there yet, but it's coming. It's on its way. Cerebrospinal fluid is basically the fluid in which the brain is soaking, right? It's the fluid literally circulating around the brain. So in many ways, it's a really important indicator of what is happening inside the brain. And it can also be a way to deliver medicines to the brain if you inject things into the cerebrospinal fluid, for example. There are also blood-based biomarkers that a lot of people are trying to develop because having a blood test is going to be a lot easier to reach more people more quickly. Of course, it's farther away from the brain, so there's probably going to be less information that we can glean, but that doesn't mean that it isn't still going to be a really important tool to that whole process of diagnosis and care and drug development. There are also skin biopsies, and that's a little bit more emerging, but it's still an important potential tool here. And Dr. Ghoshal and Drew are going to be talking about what it's like to go through each of these different types of procedures from the perspective of the neurologist and the professional and the researcher, as well as the participant who goes through them. So just really quickly, this is something that AFTD has been working on for a long time to try to really galvanize progress for the field. Across our Biomarkers initiative and our partnership with the ADDF Diagnostics Accelerator, there's $10 million that AFTD programs have put towards biomarker development for FTD over the last 7 years. It's also integral to a lot of the work that we do in fostering collaboration and conversations across different types of researchers to really catalyze collaboration and progress across the field. So when you think about the steps needed to develop these biomarkers, there's a few things to keep in mind from the participant side of things. One is that the steps that you might experience if you participate in clinical trials or in observational studies, you may go through procedures where they're gathering these types of measures from you. And they won't know whether or not it should inform your clinical care in almost all cases because we don't have enough confidence in what these things mean yet. But gathering that information and gathering those samples is key to being able to develop these biomarkers. Now, if you or a loved one is participating in clinical research, you should know that for any type of clinical research, there is an independent ethical review board that has to approve all aspects of the study before it is launched. And there are also what's called informed consent. So participants, when they are consented to join a study after they get informed about the benefits and risks of participation, as well as whether or not the collected samples and the data is going to be used just for that specific study or more broadly. So in some cases, for example, you might have extra samples gathered that they think they're going to use, but it doesn't end up getting used. And that informed consent process will usually tell you whether or not those samples might be used to advance research beyond the single study. So we're not going to get to the therapies and the care that we need for FTD without everybody working together. That's just the reality, right? This is a huge, complex problem. We all need to work together. So some things to consider for how you can help. One is consider whether or not a clinical research opportunity is right for you or for a loved one. There's a lot that's out there right now. The FTD Disorders Registry is a really important place to go to take a look at that. There's some featured studies also listed on the AFTD site. But consider if something's right for you. We also know that it may not be, right? And that's okay. It's not right for everybody to participate in clinical research. That's okay. You can stay informed about research and help to spread awareness of FTD, as well as spread awareness of those research opportunities. Because even if it's not right for you, it doesn't mean it's not going to be right for a neighbor or for somebody else that you meet in a support group, for example. And lastly, we encourage you to really join the FTD Disorders Registry, because that is a place where the insights that you share, if you join, say, the research arm of the registry, the surveys that you might answer are being used to help inform researchers when they're designing their clinical research studies, so that the values of people with lived experience and what matters to them and what types of research are more accessible and appropriate for them is something we can really communicate to researchers. So we really encourage you to join if you can. And with that, I would like to turn it over to Dr. Ghoshal. Thank you.
Thanks so much, Penny. And thanks for the opportunity to share why it is we poke and prod you so much when you participate in our research. That first slide up. Yep. So we'll be talking about those various procedures that Penny so eloquently just introduced. We'll go into the nuts and bolts just a bit, so you have a better idea of what it is that ASK is and whether it's a good fit for you or not. Next. We'll start off with something that should seem fairly familiar, the blood draw. This is something that you might encounter in a regular office visit in a clinic setting, or should you be hospitalized. Same procedure, basically, in terms of having that blood draw out of your forearm or just in the crease of your arm. The difference may be, though, in terms of the quantity that we collect, because there's a lot of bits and pieces that we're separating out from the blood, the fluid component, the cell component. There's cells there that contain DNA. So some of which is analyzed immediately, but more often we're collecting it for future use and research. So as part of that informed consent process that Penny was mentioning, that we asked if it's okay for us to store these samples longer term. So as we learn more, we can go back to these resources, your resources, and ask those finer tuned questions over time. But as such, the blood draw doesn't look or feel too different than what you might have already encountered in a routine office visit. Next. So you might say, Dr. Ghoshal, Dr. G for later on in the Q&A, That's great. So you've collected my blood. What have you already learned maybe? So as part of the research projects that Diana introduced I participate in, we've already learned a little bit, quite a bit, I should say, from those of you who've already contributed blood to our studies. We'll talk about a protein called NFL. Nope, not football, but a protein that we've come to learn in the last several years plays an important role in a lot of brain disorders, not just FTD. But here we've learned that at baseline, so the first time we meet you, we measure this particular protein in your blood, we can begin to predict based on the score or the level above and below a number, whether you are likely to go from being someone who has no symptoms to some symptoms in panel B to the full FTD clinical picture in panel C. That's exciting, right? So we talked about the importance of accurate diagnosis. Almost all of you want to know what comes next, and we haven't had great tools about that. We've been able to say that the disease progresses, it's a kind of individualized time course for each person, and now, because of those of you who've already contributed blood work to our studies, we now can start thinking about what those numbers tell us on day one that inform us about what we can expect in years 1 and 2. So that's what the importance already of a blood draw. Next. So Penny also mentioned brain imaging. So that's the business end of where FTD acts. So we want to get good quality pictures of the brain. Some of you may have had CAT scans of your brain for one reason or the other. That's using X-ray technology. So it tells us plenty in an emergency room setting, but it lacks the fine-tuned details that we can get from a brain MRI. This is a cartoon of what a brain MRI looks like, and kind of in the upper right is an image, a pretend image of type of information we can gather. Next. This is a little bit more of a real world setup for what an MRI looks like. I'm sure Drew will tell us about his experiences with that shortly. And this is, again, very important for us getting those high quality images of the brain. Next. And here's some sample images from a recent clinic patient of mine with FTD. You see some crosshairs already, and they're kind of drawing your attention to some space. So the skull can hold brain and the ever important spinal fluid. And the way these pictures are set up is if we see more black space, that means there's more spinal fluid there and less brain tissue. So there's been brain degeneration, or the D of FTD. Next. Just to make sure we're all looking at the same parts of these pictures, and the oval areas kind of draws your attention to those darker areas, or those gaps, if you will. So again, those are the temporal lobes, so the T of FTD. Next. So while the MRIs are pretty standard fare and may encounter those in your regular kind of clinical evaluations for any number of other reasons, we now have PET imaging that Penny mentioned as well. Couple of different kinds of PET images. We'll start off with talking about FDG PET. Won't get into the details of what that stands for. Suffice it to say that we're looking at how your brain uses sugar, or the G stands for glucose. So here's a little bit of a schematic of what that might look like. So we need to control the amount of sugar already flowing in your system. So we may ask you to kind of monitor your diet for a day or so before. Certainly fasting for a period of time before the study itself. Again, these are depending on which study you're in or which site you're at, they'll give you those details of what's expected of you. We check some blood work the day of so we know how much glucose or sugar is already in your system. And then in panel 4, we're talking about giving you a dose of glucose that we can see in the scanner. Okay, so it's a known amount of glucose that we're introducing to your system. It's a little radioactive, safe amount, of course, also approved by those institutional review boards Penny mentioned. And then we let that flow in your system for about an hour or so. And then another scanner that looks a little bit different than an MRI scanner. And that's where we're looking to see what is your brain's use, how is it using that glucose. So in the lower left here, I have a little balloon saying that the goal of FDG PET is to track glucose. We're looking for areas of degeneration, the D of FTD. And you can think about this like a weather map. So we're looking for cold fronts, which unfortunately a lot of our countries about to experience or has been experiencing this season. So we're looking for blue and green colors. Next. Same patient as I showed you before. So I'm going to give you a moment to see if you can find those blue-green areas. See what you think might be going on in this person. Drawing your attention then, so what's kind of in this blocked off area, what's contained within that rectangle is the blue, blue-green, mostly blue in this situation. And just below it, you might see some of those warm fronts, right? So those warmer colors of yellow, red, and orange. So in this person, their temporal lobes, again, the T of FTD, is experiencing a cold front, not much glucose or sugar being used there. That makes sense. That's a part of the brain that isn't functioning optimally in FTD, and it's not needing a lot of glucose to get that done. We have some new opportunities, though, now in PET imaging. So Penny talked about the advances in the Alzheimer's space, and we've actually been able to benefit from that in FTD as well. The one protein in common between those disorders, often, not all FTD, but several forms of FTD have tau protein as the culprit. So tau is a good protein that goes bad in the disease, and we now have ways of seeing tau itself. So do you have tau deposits in your brain? So just a similar take from the prior cartoon of this. We want to make sure that kind of start really from panel 4. We're not really needing to do too much extra preparation for this typically. But here, again, in that balloon, we're tracking tau. So if you have tau deposits, this is the injection will label that tau deposition. We're looking for warm fronts, or so red-orange colors here. We do still need time for that to circulate in your body. And again, you go through a scanner, and we're able to make those measurements. So here, we kind of bring it all together for you. Okay, so in the leftmost column, you see the MRI. The second column, the FDG PET, or the glucose labeled column here. And the extreme right, it's the tau PET. So again, let's take a look. Take a moment to get yourself oriented. It's in the boxed in area in the MRI. You can see those gaps, right? So this isn't someone with FTD in this particular example, but the gaps are important because it reflects the brain shrinkage. Next box over, we see the green, so a cold front, so not so much glucose being needed there. And the extreme right, we see the warm front because there's a lot of tau there. So this is how this technology is all coming together so we can become more savvy about identifying the right people to determine which pathologies are involved and making sure we're recruiting the right people for the right trials. The ever popular lumbar puncture. So this is a procedure that we often will refer to as a spinal tap or an LP. One of my colleagues says we have a branding problem. So she would rather that we refer to an LP as a lovely procedure. This is what we're asking of. So again, the spinal fluid is bathing your brain and spinal cord. It acts like a shock absorber. And when we sample it, and I'll go through those steps in just a moment, we're going safely, what I say, south of the spinal cord. So I know people have concerns about that, but we're going safely well below the level of the spinal cord. We may have you positioned as in figure 1 here on your side, your back, towards us, kind of curved out, arched out, or we more often nowadays will have you seated and leaning forward with some support. We'll numb up the lower back area, just that skin area. We usually use lidocaine or xylocaine, similar to the Novocaine you might have at the dentist's office rather. It doesn't deaden all the way down to your toes, but it does numb up that area that we're about to work in. Give that a couple of 2 minutes to kick in. Then we put the real deal in. You'll notice it's the pointy parts of your back. So when you arch out your back, those bony prominences, we're going to shimmy the needle between those. Again, south of the spinal cord, we're able to collect the spinal fluid, the ever important fluid that is the closest to your brain. So it really allows us to report on what the brain's doing about as closely as we can without taking a look at the brain itself. The spinal fluid that we draw should let you know you have about 150 sugar cubes worth of spinal fluid running around. And for our studies, it depends. We'll collect between 15 and 30 or so. It's a fluid that you are constantly making as a filtered version of your blood, so you make it back in just a few hours. So what have we already learned from the spinal fluid? back to our friend NFL, that protein I mentioned earlier. So it turns out, as you well know, that FTD affects younger people on average than Alzheimer's disease. But there are early onset Alzheimer's patients as well. That means that age range often overlaps. How do we sort things out? So here we looked at the NFL level in spinal fluid of folks who had no disease, FTD, or early onset Alzheimer's disease. And you'll see these squares and triangles separate out pretty nicely. So in this oval here are our FTD folks. So it's really a tool already that helps us separate out the two different populations. And again, super important when we have patients who are of a similar age and potentially clinically look very similar or at least overlap. And we want to make sure that we're recruiting the right folks for the right studies, developing the appropriate therapies that we know moving forward will be addressing the appropriate problem. One of the new up-and-coming procedures that we may ask you to do is called a skin biopsy. And here we're using something you also may have encountered in a dermatology office. So the punch tool in the center there is about a ballpoint pen length tool, maybe a little bit shorter actually. Here also we use some local numbing medicine. We're taking some skin tissue from the upper haunches, if you will, so your upper hip area, where we can spare a little bit of tissue and no scarring and things like that. It's cosmetically very minimally invasive, if you will. We collect this plug of tissue that you see in the upper right. We put it in a sample bottle that you see in the middle there to the right. And from that, we can learn a lot of information. So for instance, take that skin biopsy, we'll march across left to right. We can look at the skin cells specifically. We can kind of push them towards becoming neurons. What's the benefit of this? We now get to look at your disease in your cells and make sure it's actually reproducing what's going on in your body. It allows us then to test drugs that will work on you and your cells, and allows us to screen new opportunities, new therapies, new ways of thinking about things, about the disease, and develop therapies that we are ever more sure-footed then will work for you. So here, a little bit deeper dive with the skin biopsy then, so we can take samples from a patient versus someone who's otherwise not affected with FTD. Going through the top panel then, we can push those cells, we can look at them, we can see if we can normalize them or make them better functioning. We can see what the disease looks like in an individual. We can screen drugs that will then work for that individual. We can go through a huge library, if you will, of possible drug opportunities, if you will, that can be further developed. So these are all the reasons we poke and prod you, because we collect a great wealth of information. Now, these aren't separate from other information we're collecting at the same time. These are those biomarker and biosamples, but this is usually meshed and partnered with your clinical exam, your history, paper and pencil testing, your neurological exam, and so forth. So it's a huge database of information that we can kind of put together and make sure that moving forward, we understand the disease as kind of middle panel, disease mechanism, so understand the disease itself better, and then fine-tune the next steps towards drug development and therapies. All right, Drew, so I've kind of done the poking and prodding from my vantage point, so why don't we have you come on. And Drew has been a participant of mine for several years now and has experienced every single one of the procedures that I described here today. So Drew, why don't we do a few things? One is I'm going to talk about each procedure, and why don't you let us know what your thoughts are, your experience, what you wish you knew about each of those things. So
Absolutely.
Okay, so how about the blood draw? So that was the first one I started with. What's your thoughts or experience with the blood draw itself?
The blood draws are not too bad, obviously. I mean, it is a few vials, obviously, in your first visit. I never liked needles myself, except for tattoos. For some reason, I'm a big baby when it comes to getting blood drawn and getting shots. But honestly, it wasn't too overwhelming. Each time I had it done, it wasn't bad. I think only one time, and it's just because my veins weren't popping out at the time. But other than that, I mean, it's safe and it doesn't take too long. And before you know it, you're done and you move on to the next phase.
Great. How about, let's talk about the brain MRI. So we'll take the different pieces there. So the MRI for you, I know you've had some back issues along the way. So what was that experience like for you?
Again, I mean, it's, laying down for an extended amount of time for me. I got into a car accident a bunch of years ago, so that's why I have the back issues. So when you lay in something like that for a long period, it affected me a little bit afterwards. But again, nothing too bad. Not too much strain on the mentals. You're just pretty much laying there and relaxing until it's done. So I mean, it's a huge part for this. So I mean, it wasn't bad.
Some people talk about noise and kind of the close quarters. Do you have any recollections of those?
Yeah, I have a really bad problem with being in somewhere tight and small. Claustrophobia is terrible for me. So I had to ask the doctors when I went multiple times, I believe, each time I went, because I've been going for a long time, obviously, as you know, to just try to put my head in there and not so much my whole body. And they did it a couple times for me. And then I started gaining more courage to go in farther and farther as these years have progressed. And now I go all the way in. They put some nice music on for me, and I just, again, relax, rest in between, and then get it done.
All right. Moving on to the PET scans, we can probably talk about those together. You've had both the FDG and the tau PET. Any thoughts there? Some people wonder about, well, gosh, I'm getting an injection, so the needle component again, as well as some radioactivity. Did either of those things cause you to pause?
No, not at all. I mean, again, that's one of the easier parts for me. After the blood draws multiple times, having another needle in me didn't affect me that time. You obviously get a little sensation when it goes in, but it's nothing terrible. It's not unbearable by any means. It's honestly one of the easiest processes that I had to do each year with that, and I didn't really think about it after the first time. I think the first time you go in is the hardest time. It's a mental game. You're getting all these different tests done, within a few days, and that's what it is. But each year it got easier and easier for me to get all these different testings done on myself.
Okay. Now let's talk about the lovely procedure, the LP itself. So I should preface for the audience that Drew not only does this for us for our annual visits, but we had an AFTD sponsored study that required, the ask was pretty demanding. So we asked for a series of 5 LPs over a couple of months, and Drew did that like a champ as well. So I did kind of cherry pick him for this session to be able to tell us what his thoughts are and his experience has been with the LP.
So again, that was the scariest part for the first year or two. Being when I was younger, obviously, you always heard horror stories. Oh, spinal taps, they called them, and you got a 50-50 chance. It's nothing like that, to be honest with you. Obviously, when they numb you up, I mean, they really make you comfortable, especially Dr. Ghoshal here, because she's done, I think, all of them but one on me. It's all about comfortability and you're making yourself prepared for what's about to happen realistically. And like I said, the first time was very, very very scary for me, just because I haven't had a needle like that in my back. So I was a little worried at first, but then once it was done, it got easier each time for me, just easier. Now I don't even think about it. I'm like, okay, is it in? Oh, you're done. I mean, that type of thing. And that's as you progress, that's what you'll get into. It's like second nature, it's done and over with, and you move on to the next phase. So it's not too bad. I don't think I had any instances where something really terrible happened to me at all. You just got to make sure after it's done, you relax. And I learned that the hard way my first time. I decided to go downtown, and obviously I was in St. Louis, and go to a hockey game. What a mistake. Because I was broken down for like 3 days after that. You really need to relax after it. You get your rest, build your fluids back up in your body. Like she previously said, make sure you eat plenty of good food, and there's plenty in St. Louis if you end up going to that location. Yeah, so that's, I mean, again, it's not terrible. You might be worried about it the first time, but after that, each time gets easier and easier, and it has for me at least. So yeah.
Great. And then the last one we'll talk about is the skin biopsy. Now, Drew, I think we did that pretty early on with you. So if you can recall what that experience was like, and kind of what you wish you knew now or then that you now know.
I mean, it was probably the weirdest part, I'd say, out of anything, because it was a biopsy out of my buttocks. Obviously, it was just a little bit weird, you know, going in, taking a little dig, and it's gone. Obviously, the skin grows back. It it kind of stung for a second. It's almost like getting stung by a bee when you get that done. Again, nothing major. Didn't think anything after that. You know, it's done. It's over with. But that was probably one of the easiest parts, and it only happened once. But it was a good way to get involved with the program, and that's when I really started. I think it was 8 years ago or something. Or 7. It's been a while. Um, so and again, this whole thing is just the better of good, and that's why I do it. You know, a lot of family members and whatnot. So yeah, this is it's an amazing thing to do.
So that leaves us kind of with your closing thoughts. So yes, it's been probably 8 or 9 years, maybe even already with you.
Yeah.
Looking back now, just the whole experience, anything you wish to of your own experience that you want to share, or think might be a game changer for others out there to make these decisions for themselves.
I mean, yeah. I mean, listen, I have I think it was 8 family members on my dad's side at the time. They all passed away from this disease. You know, there was no big researchers at the time, nothing huge to figure anything out. You know, and when Dr. Ghoshal came down to where we live here and said, hey, we have some research, we can get this done. I jumped on board right away. I want to figure out if it might not save me down the road, right? It might save my kids if they end up getting it. If somebody else's family members have it, it's going to help down the road, generation after generation, if we could find some type of way to stop this, or at least curve it for a little bit longer, or what have you. That's why I'm doing it. I'm not doing it for me for everybody to know. It's not for me, because who knows what's going to happen between now and 5 years, 10 years, 12 years, 20 years from now. Hopefully we do come up with some type of solution for it. But it's for everybody. It's for humanity. That's what you got to think about when you're thinking about getting this done. Do you want to know if you have it? If you have it, how you're going to deal with your daily lifestyle? Me mentally, it disrupted me for a couple months, and then I said, "Man, I got a lot of life to live, so let's keep doing this." I sign up for everything that they have, and I mean everything. Dr. Ghoshal knows this. If they have something, I want in because I want to help. There's a lot of people like that, and there's some that are still skeptical. I enjoy doing this. It's not a vacation by any means. It's grueling on your mind, and it's just how you react to the situations that you're going to be put into when you're getting this stuff done. I enjoy doing it because I know it's going to help somebody else. That's what it's about. It's not about me at this point. It's about everybody else, and that's why I like doing it. And again, it was scary the first time, and then after that, it's a breeze. It's really a breeze, and everybody that I deal with is amazing. Not one issue. Always smiling, always joking around. It's a good time, and again, it's something that I would suggest to anybody that is interested, anybody that's worried. You're hearing from somebody that's been doing it for many of years, and I'm going to continue to do it until we find some type of solution. And I'd highly recommend, if you're contemplating on it, if you're on the fence, this would be something that I would definitely suggest to do.
Great. Thanks so much, Drew, for sharing your thoughts. Now, it is a perfect segue for us to go into the Q&A session if we're ready.
Okay. Thank you, Nupur, Drew, and Penny, for sharing your expertise. We'll take some questions from the audience now. As a reminder, use the questions box to enter the questions, and we'll answer as many of them as we can. Before I take the first question, I'll share the results of the earlier poll. 42% of the audience responded that they would definitely be interested in participating in research. 40% responded maybe, 2% no, and 16% indicated they had already taken part in research. Okay, so now for the first audience question. What kind of side effects do people experience after some of these procedures? And I think particularly because it's a recurring sort of theme, LP seems to be really the top of everybody's mind. So Dr. Ghoshal, can you please respond to that?
Sure. So I think Drew kind of put it quite well that it's a lot of it's an interesting procedure, right? So it's happening behind you. So there's a lot of anxiety I think people experience going into it. We're now talking about the side effects afterwards. Quite well tolerated, actually. We do monitor for people feeling if they're lightheaded, headachy, anything like that. There is a post-LP headache that rarely occurs, but can occur. And we don't, if and when it does, we make sure we're actually asking about it. So at our site, we keep people here an extra night. So you get your lumbar puncture. We ask you to stay in town a night. We check in with you the next morning to make sure that before you head home, we've actually addressed it, if anything. You may wonder what the answer is. How do we mitigate against that? So one is staying really well hydrated. One of the few times a doc is going to tell you caffeine is great. And what do we need to, we use a special kind of needle that really, instead of making a cut to get to the spinal fluid, it really parts the curtains, if you will, so we can get to the fluid and the curtains kind of fall back once the needle's out. Really minimizes the risk of the headache. But should you have a headache, we have a fix for that too. It's usually a blood draw that we have our anesthesiologists do, just a regular blood draw, and it's reinserting that blood into space we were just in. You can think of it like spackle. So basically, as that blood seals up, it seals up that hole, and the headache almost immediately goes away. So we try to minimize that risk on the front end. We give you instructions on how to prevent it on the back end, and we check in with you and make sure that if you are experiencing those symptoms, that we have a solution for it before we send you back home.
Okay, good. Thank you. And some individuals are actually interested in knowing whether in taking part in some of these procedures, if they're able to take their regular prescription medications, especially if they're on things that help with agitation or anxiety. Is it allowable to go ahead and continue the medications?
So I would say, as in two parts, some of it may be site specific. So there are, for the PET scans in some sites, there are a few restrictions about medications, but point being, if you sign up for it, as you're being ushered into that program at each site, they will guide you through what is permissible. Certainly for the spinal tap itself, the lumbar puncture, blood thinners are something we'll ask about for sure early and often. It is a volunteer procedure, so nothing, it's not for your clinical care. So we want to make sure that if you are going to be off of blood thinners for a few days to a week, that your doctor's okay with that and it's safe to do so. So I would say that each site may handle this a little bit differently, but they will usher you through the whole system and make sure that you are always not without your meds for an undue period of time.
Okay, good. What type of medical professionals would be associated with doing some of these procedures?
Yeah. So also potentially a little bit site specific, but I'll speak to my own statistics. So as of last year, I've done 700 LPs. Just kind of kept track for my own interest. So that's after my training years and through the course of these studies and Alzheimer's studies as well. So we have trained folks at Washington University. We call them our LP doc team. So doctors who are neurology trained physicians who are doing on the order of 30 to 50 LPs a year. So they remain, their hands are good. Other sites may have nurse practitioners, physician assistants, also well trained under the direction of physicians as well. So that's usually the LP one that comes up to mind. Blood draws are done by either phlebotomists or nurses.
Okay. And do participants typically get their own results?
$1-million question to some extent. This is coming up quite a bit. So Penny introduced the idea that these are all novel biomarkers, that we're in a discovery space. We're learning about them. We're understanding the power that they may have for us moving forward. It means we might be just short of being able to have a lot of confidence in being able to say, guess what, your tau is 120 today. And you're going to say, so Dr. G, tell me, what does that mean? Do I have it or not? I don't know. Is kind of where we still are. So the graphs, a few data that I showed you were kind of big groups of people and group analyses, and we are learning a lot. So we're not quite at the point, it's not quite ready for prime time, but we are certainly interested in making sure, and the surveys that you guys are filling out and saying, hey, I really am interested in hearing about my data, are very much informing how we can do this responsibly. So when we let you know something, I think it's a little bit easier to do with an MRI, for instance, than with a blood or CSF biomarkers just yet. But we are interested in moving in that direction for your benefit.
And can people in later stages of FTD actually take part in research procedures?
Absolutely. So in fact, one of the studies that you, Diane, actually introduced me as being part of is all FTD. So in this last go around of funding, we had what we call the biofluids arm of the study. So it can be a one and done type of thing. So it's a little bit of expanded version. You have your regular clinic visit, blood draw with that, some surveys, so you continue to be able to contribute to science, but it's not the full ask of the MRI and the spinal fluid and so forth. But if you, it's also just as easy that as you get to know us and say, oh, I think I would like to do more, we're able to do that as well. But yes, we are mindful that at advanced stages of dementia, that may become more challenging. So the ask is kind of measured accordingly.
And I'm going to flip the question. Is it also important to have early stage and maybe even healthy controls participate in some of these procedures?
Absolutely. Healthy controls, absolutely. So we need to know these are some good proteins that are acting awry in these conditions. So we need to know kind of what in normal population, what those numbers can look like. So we can understand where it is that with FTD we land. So certainly, so far the way we've done that in all FTD in particular is we don't ask for people to need to know their own genetic status. You do not need to know that you do or do not carry the family disease, the family gene mutation, rather just that there is a family history from one arm of the study. And that means your siblings, so when we ask you, you don't know just because we asked you, if you, oh, that must mean I must have it, right? So we really come and ask you and your siblings, cousins, so forth, and that way we basically have some controls amongst you. It's actually very informative, is how we did the Tangle study as well.
And can participants choose which procedures that they're willing to do?
For the most, yes, the informed consent means you get to let us know what it is that you will choose to participate in or not. I think the ask is always the whole ask, at least at our site. The one that some people I think toggle with and kind of go back and forth is a lumbar puncture. Usually the blood draw doesn't seem to be the hurdle nor the imaging protocols, but the spinal tap may be the one that cause people to pause, and that's fine. That's kind of accounted for across studies of this nature. But we, at least at WashU, we ask you to step into the full space.
Okay. And I'm going to squeeze a quick one in here, but this will be the last question. But this really refers to, can samples only be used for the current study that individuals taking part in, or is there some level of future or data sharing use as part of studies?
Absolutely. You are contributing to a large repository where we're able to, it's part of the informed consent where we ask, is that okay for us to do? And then yes, your samples are under lock and key, but they are shared with approval amongst researchers both in our consortium and outside when the appropriate kind of scientific rigor is met. But yes, it can help contribute to ongoing and future other related studies, yes.
That's wonderful. That's very important to know. Okay, that's all the time we have for today. Thank you, Penny, Nupur, and Drew for being with us. We hope you found the presentation to be informative. And as a reminder, the webinar has been recorded, so you'll be able to find it both on the AFTD and the FTD Disorders Registry websites and YouTube channels in the coming days. Before we leave, I want to revisit the polling question and ask you to respond again now that we've shared today's educational information. Click on the answer that most accurately answers the question, would you consider taking part in FTD research in the future? And we'll give you a couple seconds to answer that. All right.
And I can jump in with the results here.
Okay, great.
This is Matt from AFTD, and we're up to 50% people said yes, 26% maybe not sure, 2% said no, and then 22% I've already taken part.
That's excellent. Thank you, Matt. Okay, and let's go ahead and introduce you to what the next webinar will be in the Perspectives in FTD Research series. and that is entitled Amplifying Your Voice in FTD Research. Why am I asked so many questions? And this is scheduled for March 28th. Look for communications from AFTD and the FTD Disorders Registry in the coming weeks for information on how to register. Thank you for joining us today. Let us know if you have any comments or questions about today's presentation. If you have any further questions about FTD, you can contact the AFTD's helpline at 866-507-7222 or by emailing info@theaftd.org. And be sure to sign up for the FTD Disorders Registry if you haven't already. Visit ftdregistry.org to learn more. Thank you so very much for attending today.